So the next speaker is Saul Villeda. He's from University of California, San Francisco, and the provocative title of his talk is Mechanisms of Brain Aging and Rejuvenation.
Hello, everybody. First of all, thank you to organizers and to Molly in particular for the invitation to come and speak to you today. And broadly, I do want to talk about this idea of reversibility. And broadly in my lab, we're really interested in addressing the question of whether the effects of aging, particularly on regenerative and cognitive function, can actually be reversed. So can we take an organism that's already old, it's gone through the natural aging process, and somehow reverse some of those effects, even at a cognitive level? Now, I think that this could really be a transformative way of thinking about things in that, during normal aging, of course, we have this decline in at least some aspects of cognitive function, and a lot of it really associates later in life with this susceptibility, vulnerability to neurodegenerative diseases. However, if we think about it, during the youthful state of our sort of body or in an organism, there's some way that the brain can naturally counteract some of these insults that are coming from the environment. So there's something about this youthful state that can actually combat the effects of aging. The idea here being, is there some way of restoring that youthful state, at least in part, within the aged brain? And if so, can we extend what Steve sort of called health span, right? Can we extend the amount of time that we have sort of this integrity still there? Not lifespan, but rather health span. Now, in particular, my lab uses mouse models as a way to sort of start looking into this idea of reversibility. We can age mice naturally, ask in animals that are cognitively impaired, are there ways of improving them? Are there ways of restoring this function? And in particular, we use tasks that are both associated with regenerative capacity, stem cell function, as well as tasks that show age-related impairments. And one of them being spatial learning and memory. So I love to give this example. I know that most of you will know this, but I still love it because I get to show the picture of the Tesla. So I'm from the Bay Area, and everyone in the Bay Area now is driving a Tesla, except for assistant professors such as myself. But let's pretend that I can't afford a Tesla, and my Tesla is parked somewhere in there. Of course, I'm going to come in earlier than all of the full professors to work, and I'm going to park my Tesla, and I have to remember where it is, right? So we're going to have these spatial cues around there. We're going to have, let's say, the Golden Gate Bridge or a really bright rainbow flag and my favorite taco truck, because that's what's in style in San Francisco right now. So similar to this, I'm going to be able to see these cues, and it's going to guide me at the end of the day to find my Tesla, and if I could afford it, it would be red so everyone could see it. So this similar task that we have, right, this idea of spatial learning and memory, I'm guided to this car based on these cues. As we get older, this does decline, right? And I love showing this to sort of my parents, and that they're the ones that are now using a car alarm to start finding their car. So I tell them, Mom, that's your brain on aging. We can use something very similar, right, in rodents, which is just a pool of opaque water, a hidden platform instead of a Tesla, and some cues around a room instead of a bridge and a flag and a taco truck. We can use these bright things. And we can teach a young animal how to find it. Again, it's a very basic task, but it still gives us some power in terms of looking at both what's driving impairments and is it possible to reverse them. A young animal obviously learns, this task very well. An old animal will learn the task, but it is going to be impaired. It's going to take either longer, make more errors in finding it. Now, this is a really basic experiment, but again, we can go both ways. We can look at what's promoting aging, what's reversing aging. Now, in particular, this is governed by the hippocampus. This is just a cross-section of a mouse brain. We're going to zoom in really quickly, and here we have the trisynaptic circuit of the hippocampus, right? It's physics, basically. However, the neurons don't function on their own. The hippocampus is a really unique region. Number one, it still has regenerative capacity. There's neurostem cells and neurogenesis that still occurs. Additionally, we're appreciating the influence of other cell types. For example, the resident macrophages, or the microglia. We now know that they're really important for maintaining homeostasis and maintaining that integrity of the neurons at the level of the synapses. Now, with aging, there's a number of different cellular changes that happen. We lose regenerative capacity. We increase the activation of these microglia. They become inflamed, and people even think that they may be contributing to synaptic pruning. And collectively, this is then going to change the synaptic dynamics of the old brain, and we're going to cause functional decline. Now, importantly, all the players are still here. The microglia, the stem cells, the neurons, synaptic plasticity, it's there. However, it's declining. There's a dysfunction. And the idea is, if all the players are there, can we restore at least some of these cellular hallmarks back to what they look like in the younger brain? And if so, does that have a functional consequence? Now, in thinking about aging, we really took a step back and we thought, okay, aging doesn't just occur in the brain. It occurs throughout the entire organism. So let's then look at an approach that also is going to look at the influence at an organismal level. And we thought, what connects all organs together? What connects all tissue together? And the answer was blood. So we wanted to know, could blood have an influence then on the brain? And for that, we use this really old surgical technique known as parabiosis, in which we physically connect together two animals, such that old blood is flowing into the young, and young blood is flowing into the old, two hearts pumping one bloodstream. And we can ask, are there pro-aging drivers in blood, or are there pro-youthful drivers in young blood? And we tested again compared to isochronic young or old. Now, the first question we address is this idea of pro-aging factors. Are there pro-aging factors? Can we target them in old blood to somehow restore function? Now, in order to do this, you first have to identify factors. So we did a targeted proteomic screen, and we identified immune molecules actually as very prominent in the old blood, very prominently associated with decreased neurogenesis and increased inflammation. I'm going to share with you one factor. However, this is, again, sort of a broad story of how inflammation at a systemic level may be driving a lot of this cellular change. Now, in particular, we identified what's called B2M. It's part of major histocompatibility complex class 1. You'll know it probably mostly in tissue rejection. When tissue is rejected, it's due to these MHC1 molecules. Now, traditionally in the immune component, they're used for antigen presentation to surveying T cells, so it has a very canonical immune component. During brain development, however, this idea, again, of developmental processes coming back and aging, during brain development, it's involved in synaptic pruning. The same molecules, completely different role. So the idea was, could it have an effect then also within cognitive function, within regenerative capacity? Now, the first thing we did is just, again, confirm that there is indeed an age-related increase in plasma. We also looked at our parabiosis model, and we saw that there's an increase in blood levels, also after exposure to old blood. We then went and we had access to human samples, and the reason we were really excited about this is in human patients undergoing chronic hemodialysis, there's an increase in blood levels of B2M that correlate with cognitive decline. HIV patients that develop dementia also have an increased level of B2M, and it's also in cerebrospinal fluid of Alzheimer's disease patients. So when we looked, again, we saw an increase both in plasma between 24 and 92 years old, and also when we looked at a more modest number of cerebrospinal fluid samples, we see this increase again. So again, we could go between the mouse and the human to see whether there's some relevance behind it. Now, to get at functional assessment, which we can do in mouse models, we then ask what happens if we mimic the systemic increase. We pharmacologically increase it and then follow by behavioral paradigms. We do both contextual fear conditioning and water maze, but I'll just show you water maze data. Here I'm just showing you histology of double corin. These are newly born neurons within the hippocampus. You can see exposure to this B2M decreases neurogenesis. We do a shorter task of the water maze known as the radial arm water maze, where we divide a pool of water into a series of arms. The reason we do this is it has a robust age-related decline, and it allows us to do the task faster and use less sample size. And you'll see for the second part of the talk why that's important to us. The first day, we alternate visible and hidden platforms, and then the last day, we have only hidden platforms. And here you can see that we have the vehicle versus beta-2M treated animals. Again, this was intraorbital, so it's in the bloodstream. When we come back the second day, you'll see a separation. Animals receiving B2M commit more errors. They have a harder time finding this platform. Additionally, we saw that increase in cerebrospinal fluid, so we went into the mouse model and asked, is there an increase in the hippocampus? And here I'm just showing you a Western blot showing an increase during aging of B2M. We looked at our parabiosis model and asked, exposure to old blood, does that also increase B2M in the hippocampus? And it does. So then we wanted to know, well, what happens if we mimic now this effect locally? And for this, we did stereotactic injections, where we can do either unilateral control versus beta-2M, or for cognition, we can do bilateral, straight into the dentate gyrus. Now here I'm showing you the same section on contralateral sides of either vehicle or B2M. And you can see local delivery decreases neurogenesis. When we do then water maze, again, animals receiving B2M locally have even greater number of errors that they commit versus vehicle. Now, having observed this, we wanted to know, well, if this is going to be a molecule that we're interested in, then are the effects of B2M permanent or are they reversible? If you're trying to target something at old age, then the effect can't be permanent. If not, the system can't recover. So what we did is we stereotactically injected B2M and waited longer. We gave it a month to recover, 1 full cycle of neurogenesis. And what we see is that those animals no longer show those cognitive impairments, again, suggesting that this molecule has an effect, but that effect can be reversed. Now, lastly, having observed both an increase in the blood and increase in the brain, we wanted to use genetics. And we used a whole body knockout to get rid of B2M everywhere. And we did this at both young and old ages. Now, interesting, when we look at a young animal, they're absolutely normal. We see nothing wrong with them, at least at the level of neurogenesis. However, when we look at an old beta-2M knockout, you actually see an increase in regenerative capacity. When we looked at cognition, similarly, a young animal has no change in cognitive function, either by water maze or fear conditioning. But when we look at this old knockout, you can actually see that they commit many less errors than a normally old animal, again, suggesting that decreasing or eliminating this immune molecule can actually somehow counteract or prevent some of this impairment that we normally see later in life. Now, again, this is just a snapshot. We've gone on and done a lot more mechanistic work and also combined it with parabiosis. But again, getting back to this idea of can targeting pro-aging factors, is that one way of rejuvenating? Well, I haven't shown you that. I've shown you that we can target genetically these molecules and at least prevent. And ongoing work right now is actually using small molecules and antibodies to actually see, can we target this even at old age and do something functionally either to regenerative or cognitive function? Now, that's one side of the coin. My lab is also looking at the converse, which is, can pro-youthful factors in young blood rejuvenate regenerative and cognitive function? Now, over 10 years worth of work has actually shown that exposure of an old animal to young blood can actually enhance stem cell function, both in muscle, in bone, and even in brain. And we've shown that, and Lee Rubin's group has also shown that at Harvard. However, one question we were really interested in is, can it enhance other forms of brain plasticity? You can think about stem cells. Stem cells are going to give rise to newly born neurons, which means those cells have not experienced aging. They're being introduced into an aged environment, so they may be more plastic. But what about neurons that have been there the entire time, 18 months worth of age? Can they somehow also be affected by young blood? And for this, we specifically looked at synaptic plasticity and structure, dendritic spines. Now, what we see is during aging, what we see is they're both more dynamic. However, when you take a snapshot such as the Golgi, which I'm showing you here, you actually see a decrease in dendritic spines. But exposure to young blood through a model of parabiosis actually increases the number of dendritic spines. So even those neurons that are there the entire time actually have an increase in dendritic spines. We then did behavior. This time we did intravenous injection. We did 5% of plasma by volume of the animal, 8 times over 24 days, followed by behavior. And again, all of these were litter mates that were either given age-matched plasma or young plasma. And what we saw here is the animals here in gray, you can see given age-matched plasma make many errors. However, litter mates that were given young plasma make many less errors. And again, this has actually been observed across institutions. So something about young blood can actually reverse some of the cognitive impairments that we're seeing in an aged animal. Again, suggesting that there's malleability even later in life to sort of restore some of this function. So again, going at these, can pro-youthful factors then rejuvenate? We believe that they can. We believe that right now the search for a number of different factors is going on. I believe there's some articles that will be coming out soon from a number of different labs that are addressing this and trying to identify individual factors. The consensus is really that it'll be a combination, but it's still really exciting to think about the fact that even at old age, we can still have interventions that can improve some function. Now, ongoing directions for us is really understanding the contribution of the different cell types. You know, the brain is not in isolation. It's sort of an additive effect of all sorts of different cells. So we're looking at the stem cells, at the microglia, at the neurons. We're looking at molecular changes, both transcriptional, post-translational, epigenetic, looking at other types of cognitive impairments that occur in other regions. Are they reversible? Is it specific to particular regions of the brain? And just sort of moving forward, I think this idea of reversibility is very unique. This idea that even later in life, in the absence at least of gross neurodegeneration of cell death, there's still some plasticity, some malleability left in the aged brain. And I think it's a different way of approaching cognitive aging and asking, there's ways of preventing, there's ways of sort of tapping into what may already be there. And I think this is one way in which we can start looking at other components of the body, such as blood, the systemic environment, and how that's influencing the brain. I think other interventions such as exercise and caloric restriction all go along the same line, which is how can we use what's outside of the brain to understand and even cause interventions within the brain. And with that, I'll thank my graduate students, Luke, Cedric, and Chris, who did a lot of this work, and of course, our funding. Thank you.
Yes, questions?
Great talk. Can you comment a little bit more about the mechanisms through which the beta-2M might be leading to these changes? I mean, at least in the clinic, many times we see the beta-2M increase, including in aging, associated with other conditions such as inflammatory conditions. So, I mean, I know that you did some direct injection experiments, but to what extent it might be mediated via inflammation, or do you think it's a direct effect of beta-2M and genes involved in neurogenesis, et cetera?
No, great question. I actually think you're almost getting a double whammy during aging in that, yes, there's increased inflammation. We've done some studies, very preliminary, where we do heterochronic hematopoietic stem cell transplantations, and we see that a lot of the beta-2Ms actually derive from the hematopoietic system itself. So that's one thing that's driving it. The other is that it does have a direct effect, and it seems to be working in that complex. I said MHC1s usually with beta-2M together, and it seems that they stay on the cell surface and they serve as brakes. Just like in development, you have this complex and it signals synaptic pruning. In the old brain, you have this complex and it signals stop proliferation or increase activation of microglia. So it's the same developmental program, but you're upregulating in a context where you don't want it.
Thank you.
Yes, and, this gentleman here in the blue shirt.
Okay. Great talk. I'm curious also about the mechanism. When you say that you increase neurogenesis by taking away beta-2M, are you actually increasing proliferation, differentiation, or survival of those new neurons?
Yeah, so what we're doing is it turns out that there's over 50 MHC1 molecules that are thought to be expressed in the brain, so there's a number of them. One of my graduate students had now pinpointed it down to two classical ones, the classical HLAs, or H2K, H2D in the mouse model. And it seems to be working similar to natural killer cells, where these complexes are on the cell surface and they actually sequester growth factor receptors, such as FGF receptor, EGF receptor. So what seems to be happening is the MHC1s on the stem cells sequester these growth factors and prevent proliferation from happening. Subsequently, that leads to less neuroblasts and less neuronal differentiation, but it seems to happen very early on. You release the brake, all of a sudden you have an increase in neurogenesis, even in the context of aging, and we've been doing some genetic studies to do that. Yeah.
I was curious about when you showed the data where beta-2M changes with aging in humans, do we know that actually helps differentiate whether the patients are getting dementia or cognitive deficits? Is it predictive, or is there any association with that?
Yeah, that's a great question. So I've had the opportunity to look at samples at the memory clinic, but it's very limited in sample, so it's all preliminary. It works best in people that already have Alzheimer's disease. So it works as sort of this panel of biomarkers. It can be one if used with another, let's say 18 markers that seems to at least be able to segregate. I haven't looked at, unfortunately we didn't have cognitive scores for these particular samples. It was actually really hard to get this many samples for us. But I do want to see that. I know that there have been studies that have been done particularly in, I believe, HIV-related dementia and chronic hemodialysis, where they did do that and they got a segregation. I don't know about prediction, but they got a segregation between them.
One last
Well,
question.
You just blew my mind with the vampire rodents, right? Is there, just curious, is there any, have there been studies about blood transfusion and temporary cognitive improvements, getting like young blood into older adults?
So I get that question a lot. Unfortunately, whenever there's, oh my goodness, am I over? No. Unfortunately, whenever you do blood transfusions, it's in the context of something else. So there's comorbidities or some sort of injury or disease. So there's nothing really clear. There are clinical trials now that are ongoing. I have mixed opinions about them, but there are ongoing trials. Some of them are very controlled and they're efficacy related. Some of them charge $8,000 to give a person an injection of young blood after the age of 35. So go figure. But people are trying at least to move forward. The early ones that are being done, I think the ones that are being done properly are right now at the stage of sort of feasibility, efficacy, that kind of stage. Yeah. Thank you.